Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is February 26, 2026, and we're talking with Dr. Peter Fox. Peter is the founding director and current director of the Research Imaging Institute here at UT San Antonio. UT Health San Antonio is what we are saying right now. And he's also a professor of radiology, neurology, and physiology at UT Health. He's a pioneer in the use of brain imaging techniques and their applications in humans and animals for research on the structural organization of brain function and for diagnosis and treatment of brain diseases and for brain stimulation as an experimental tool and also as a treatment. Hi, Peter, and thanks for joining us.
Yeah, thank you for having me.
And also with us is George Perry, professor in the Department of Neuroscience, Developmental, and Regenerative Biology. George is a brain disorders expert and a podcast veteran. Hi, George.
Yes, thank you for having me.
And I'm your host, Charlie Wilson. So, Peter, we often hear about we know about well-known neuropathological changes associated with specific brain diseases, like the loss of dopamine cells in Parkinson's disease, or thinning of the cortex in Alzheimer's disease, or plaques and tangles. They're really key for diagnosis and for guiding research in the disorders that have those. And they've been very beneficial, but there's a whole host of other brain disorders, or at least psychiatric and neurological disorders that don't necessarily have an obvious brain pathology. And that people have long said, well, this is a disorder of behavior and cognition, and maybe it certainly has some brain substrate, but it isn't just a loss of some brain area. So that's always seemed to me strange, that we can't see something in the brain that goes with that. And there are even other things, like great skills that people have. Apparently, neurologists examined Albert Einstein's brain and found it to be perfectly normal, like yours or mine. And that doesn't sit very well with me. It seems to me Albert Einstein's brains has got to be something special. So you have worked on, I think that one of the reasons that we have this idea is we just don't have very sensitive ways of comparing people's brains, quantitative, systematic ways of finding the subtle differences in brain structure and function that goes with each of these disorders or incredible gifts. And you have worked on refining the methods for seeing that kind of stuff. And you have seen it in places where it hadn't been seen before. And I wonder if you could sort of outline the strategy used to detect subtle changes in brain structure and physiology in human brain imaging that could be used for understanding diseases and for diagnosis and for designing treatments.
Yeah, I'd be glad to talk about that. There's been a steady progression of improvement of the methods that I've been working with over the past 30 or so years. You have to realize that the tools that we use now pretty routinely, PET scanning, MR scanning, CT scanning, didn't exist until the late 1970s and early 1980s, and were Nobel Prize winning inventions, right? So the inventor of CT, Hounsfield, won the Nobel Prize for that, and the inventors of MR collectively got a Nobel Prize for that. So in creating these new techniques, we got a set of tools that really came online in the early 80s that nobody knew exactly how to use. And so it's within that emergence of this new set of tools that we've had to try and develop them and invent the methods for finding these abnormalities. So one of the things that the distinction you need to understand is that clinically, people that are examining images, radiologists that are examining images, are looking at them one by one and doing a visual inspection, the way a doctor might examine you and see how you walk, how you look, is everything in place? So they look at brain images in the same way. And the abnormalities, even for pathologically well-known diseases, so Alzheimer's disease has clear pathology, plaques and tangles, that is not readily evident on a scan unless you have a radio tracer that's specific for the proteins for plaques and tangles, right? So we had PET scanning, but we didn't know what to label, how to label it, what the underlying pathology was. And so it was after we knew what that amyloid was in plaques and that tau was in tangles that we knew enough to make a radio tracer to then let us see it, right? So the pathology informed the imaging. So if you don't have a good understanding of the pathology, you may not even be able to make an imaging technique to see it. So if you ask, how do we see things that aren't pathologically evident? Yeah, that's kind of a tough one to break. If you don't know what you're looking for, how are you going to find it? So the methods have to be built to confirm and expand on what you know about. So we can get back to the issue of personality and intellect, but those are asking the most subtle questions. So the work that began very early actually started in Denmark with single photon tracers, so not positron emitting, but single photon, having people do speech tasks, hand movement tasks, and mapping these areas which had been known before only by lesions. If you produce a stroke, where does the stroke paralyze your hand? Where does it paralyze your foot? Where does it paralyze your leg, like your face? What is it? So we had data like that. It's very crude. And so the earliest functional brain mapping techniques were focused on mapping in real high resolution and good quality what those functional areas are, because those really weren't known. And so those techniques were developed through the 80s, the 90s. I spent a lot of time modifying those techniques and developing those. The importance of that is that now when we look for abnormalities in other diseases, so in psychiatric disorders, we know that psychiatric patients often have disorders of higher cognitive abilities, of executive function, but if you don't know where the executive function areas are, how are you going to make that function structure association? It's very difficult. And so a lot of work, decades, has gone in to very fine-grained mapping of what the functional representation in your brain is of cognition, emotion, perceptions, different perceptual modalities. So that's been a lot of the groundwork that was necessary to begin studying these higher functions and higher capabilities.
Could you just remind us how I do that? So, if you're looking for the hand area of the motor cortex, and you have a method that allows you to see activity in the hand area of motor cortex, and I guess like functional MRI would be a method like that, then you could ask me to like make a fist and over that, and then you could see where that signal comes from. And if you want to know where I make decisions, where in my brain I make decisions, I guess you could image my brain while I'm making decisions. So you have to design a decision-making task for the person to do, and for anxiety and for other emotions.
Yeah, that's exactly right. That you create states. Typically, the best data is where you have a within subject design where they're doing the task, often for a minute on, a minute off. With the early studies with PET, you could do an acquisition for a minute and then you'd have to wait 15 minutes for the tracer to decay, and then you do it again. So you can do, in a few hours, you could do, say, 10 or 12 on, off sessions. So in a session, you could have somebody move their tongue, move their hand, move their toe, and map motor cortex. So you can do that. So that was done. The same thing for vision and for auditory, that each of those got mapped, right?
And it sounds like just one trial.
So typically it's multiple trials per person per session. And then to get really robust maps, you want to do a whole group of subjects and see the individual variability and how concordant they are across subjects, and find ways of representing them all in a standard way, so against a standard brain. And so those techniques were developed earliest by neurosurgeons who were doing stereotactic surgery, where they put somebody in a modifiable frame with coordinates, and they were doing surgery with that.
Like the Penfield type experiment.
Even
more refined.
Yeah, pre-Penfield.
Oh, pre-Penfield.
Pre-Penfield. But the doing recordings for epilepsy, doing deep lesion placement for Parkinsonism. So putting in selective lesions. So that framework has been around for more than 100 years, and it was known to be reproducible across subjects. And so that's been adopted by imaging as the reference base. And so when we do examinations across people, we put them into the surgical framework and report all the data in that standard space. So that creates a body of knowledge that's all mutually referenceable, so that you're building up through this collection of studies now, many tens of thousands of studies representing normative brain structure and function.
So that was developed prior to the imaging in
Yes.
standard space. Okay.
Yes.
But how hard is it to put them into a standard? I mean, I could measure how far it is from bone, some bone landmark in each direction, but how do I know that I'm in the same part of the brain, because brains are slightly different from each other?
The dimensions and how we normalize brains to one another has gotten progressively more sophisticated over time. And you can quantify its accuracy fairly well by having different people do the same task, right? And if, as you said, well, we could do it all in one session. What if I had you do enough data so that in an individual subject I could identify the representation of the thumb or the tongue or the eyes, and then you can look across people to see how variable they are when you put them in your space. And you can test pretty readily that if you improve your spatial registration, that the results get closer to each other. So you can optimize your methods in a very straightforward way of bringing them all into registration with one another.
Use brain fiducial marks like
the early you use total dimensions of the brain. The anterior commissure is in embryogenesis, a rotational pivot. So it's relatively stationary during embryogenesis relative to other areas of the brain. And so that's used as the anchor point and the origin of the space. And so everything is defined in coordinates relative to actually the plane as the anterior posterior commissural plane, with the anterior commissure as the origin.
So these are two white matter tracks, one that goes...
That go between hemispheres. Yeah. So commissures are connecting hemispheres. Yeah, so that's used as a standard. The neurosurgeons that already worked in that space, we began working in the space early on, and then that's become very widely adopted. At this point, that I would say in all of neuroscience, that's the most widely adopted standard. I don't know if in animal studies, if there are common standards that are as robust, but there's just a massive literature that's all in that space.
And if there are, like when I was in neuroanatomy class and we were looking at 3 or 4 brains, I could see this person's sulcus was slightly different place from the next person's sulcus.
Yeah.
Does that kind of, how does that get worked back into standard, or do those differences get left?
There are warping algorithms that focus on sulci and gyri, and that's okay, and that improves normalization to some degree. But even very early on, when Brodmann was doing cytoarchitectonics, he noted really, if you ever read the original book, he's pretty clear that in some areas the sulci and gyri clearly predict cytoarchitectonics, but that's really primary sensory and motor areas, and when you get beyond that, the gyrification doesn't. And so even, say for hand motor area, it's very clearly predicted by the central sulcus, but the central sulcus ends before you get to the mouth motor area. And so mouth is not predicted by any sulcus, but it's well predicted in coordinates because it's in the same spot, even if there's no gyrus.
That's great. That's the stereotaxic coordinates are primary, and all the folding is secondary.
You can improve registration some if you incorporate sulci. If you add that additional measure in. But is that what you need everywhere? It's like, no, the sulci are more or less random when you get away from the primary sulci. And so they're more a nuisance than they are a solution.
Mm-hmm. So these brain images get used for several different purposes. One of them is just, I'd like to have a structural image of this brain because I'm going to put an electrode in the globus pallidus and I want to find the globus pallidus. Mm-hmm. One of them is I'm trying to find out a functional region in the brain, and so I have somebody perform a task. Another one is I'd like to know what the connectivity between brain regions is, and imaging is used for all 3 of those kind of things, and maybe there are more.
Yes.
And so what are the sort of variations? For example, for looking at connectivity, what's our preferred way of telling when 2 brain areas are well connected?
So there are both structural measures of connectivity. So if you're doing postmortem anatomy, you can dissect along white matter fibers. I mean, classical way is like with orange stick dissection, you can just tease the fibers apart, right? So there's an imaging analogy to that, which is using diffusion. So water molecules always, right, they have Brownian motion. And if there's an axon, the motion is constrained more along the axon than it is across the axon. So you can quantify that. And you can follow axons through the gradient of motion, the dominant gradient of motion, and really of Brownian motion. And so you can use that to define tracks and between one area and another. And so that can be used for research. That can also be used for surgical planning. So if a surgeon is planning on doing a resection that's, say, near the optic radiations, right? And so he doesn't want to disrupt a person's vision, that you can do a visual task. So give a visual stimulation and see the visual cortex, and then have a diffusion tensor image and seed it starting at that location. Say, if I follow the diffusion gradient, can I follow it back to lateral geniculate? And the answer is yes, you can. And you can define that entire structural pathway, and that information, it can be given to neurosurgeons who use the stereotactic system to help them define the zones of their surgery.
So there are a bunch of different scanners for doing these things, or you do it all in one machine?
And an MRI can do that. It can do this normal structural imaging, so to see a tumor, to see a lesion that you're resecting, and it can do the connectivity there, the tractography. Functional MRI can do functional tasks. So if the surgeon wants to know which hemisphere is language represented in, is it left or right, and when I'm doing my surgery, am I near Wernicke's area or not, that all of that can be done in the MRI scanner.
And you do that near the surgical suite or is prior to?
Prior to. You do it prior to, and then this whole notion of putting things into a coordinate space is referred to as stereotaxy.
Mm-hmm.
And so that's been around for a long time. And so the FDA has a device code called a stereotactic instrument. And stereotactic instruments are used to guide activities within brain space. And so there are neuronavigation systems intended for surgery that will get images beforehand. They go to the surgical suite. They're registered to the patient on the operating table. And as the surgeon is moving, it can tell him where he is relative to these images. So those are techniques that have been evolved over time. Actually, this personal story, when I was a resident and a fellow then at Washington University, I worked a lot with the head of neurosurgery, Sidney Goldring, and he did both epilepsy surgery and some tumor surgery. And at the time, my mother had a tumor resected, a meningioma, but had persistent seizures. And so she came to Goldring to get treated. And Goldring asked me, since I was doing epilepsy mapping with him, if I could do pre-surgical mapping for him. And so we did, using these tasks that I'd been working on. And so to my knowledge, that's the first pre-operative functional mapping done on anybody. And so that was in the Journal of Neurosurgery in 1987. And so the images in there are of my mom. So.
Well, there's when we do this kind of stuff, we usually think, well, I'm looking for the brain area. If I move my finger, where's my finger brain area? But there isn't really one brain area that's responsible. Usually there's some kind of a circuit, collaborative interaction among brain areas. And this has been a struggle for neuroscientists in general, because our brains, when we think about our brains, we start looking for centers that do things all by themselves.
Yeah.
But in watching a brain of a person doing a task, is there a way to find the cooperativity among brain regions and make some, intellectual step beyond just thinking about single brain regions that do things by themselves?
Yeah, they're really there are a number of different ways. And one of the big advantages of current imaging techniques is that they image by default the whole brain. And so if you tap your fingers, we see activity in premotor cortex, in supplementary motor area, we see cerebellum, we see basal ganglia, we see that it is in fact multi-regional. And if you test connectivity, they are all functionally and to some degree structurally connected. It's a little bit harder to get all the structural connectivity. But yes, they're working as a network. It's cooperative. And so there's been really for a long time a an understanding that brain function is network-based. I mean, even going back to very early studies with Broca and Wernicke, that the diagrams they were making based on lesion data were network diagrams. So they were discovering that you need to disconnect regions to cause abnormalities. It's not just a lesion that one area does one thing. It's that complex functions are the interaction between more than one area.
How does this play out in memory? Remembering things in imaging.
So
I'm talking about long-term memory.
Yeah, so that's a tough thing to answer. That's really not a very
I thought it had
easy.
been answered that people see things lighting up, much like when the original memory was done. Or is that the too simplistic view?
No, I'd say that is certainly true, but how the wiring works and exactly how memory is encoded. I don't know if you remember people talking about grandmother cells, and that they were very sure that you were changing the DNA in individual cells to incorporate that, like encoding that as a memory.
Halle Berry cells too.
Yeah, and that so we know that medial temporal lobe, hippocampus, amygdala are very much involved in the encoding process.
Yeah, the encoding, I think we understand more, but what about long-term retrieval?
So as you retrieve specific images, and by modality, you see brain areas lighting up. As you have people recall things, you can see that perceptual areas are responding. And so that's true, that visual imagery without a stimulus present, just internal imagery, calling up images in your mind of different objects, activates higher order visual areas. That's for sure true. But if you ask me to explain what the circuit is and how it's recreating memory, that's a pretty difficult question. That dorsolateral prefrontal cortex is robustly activated by holding something online where you're doing working memory. That's good. That's clear. But then how do those all interact? I'd say that's...
And then the next level is, how does that generate consciousness? Or all these processes. Is there anything of imaging helping to define consciousness, aside from the brain being active?
By conscious, you mean the opposite of unconscious?
Yeah, I guess. I'm not even sure I know what consciousness
use that word
is.
to mean a lot of different things.
Yeah, I'm not sure I know what it even means.
Yeah.
Unconscious, I think we know. Okay.
So certainly,
I just wonder if you could see that by imaging, get some insights.
You can definitely get some insights, but I think you need to be careful about whether the terms you're bringing to bear are scientifically based or not, and whether they're scientifically justifiable, valid questions to answer. And so, can we identify regions involved in level of alertness, right? So moving from sleeping to wakeful, right? So are you unconscious when you're asleep? It's like, well, you've got something going on. If you're dreaming, you have some consciousness, but it's not well linked to your state of self-awareness and your continuity of experience over time. So, I mean, for human consciousness, I assume animal as well, there is a continuity that it's over time, and you can remember areas of prior time, and you know they're prior, and you can envision future areas, and right. So there's a continuity of self-experience. How that's all maintained and how it's interrupted with anesthesia and then returns, and it's uninterrupted. It's like, okay, so that's a pretty amazing thing. How that works and what's establishing that is not altogether clear, and it's not really obvious how to ask questions about that. People have made assertions with imaging that I think were ill-judged. So you can make decision tasks and have people make choices, presented a forced choice, right? And so if you make a choice, it's like, well, that's free will. You're making a choice. Do you want this or do you want that? Is it a this or is it a that? And so so it turns out that there are certain areas that are very reliably activated when you're given a discrimination and you make a discrimination, right? So is this letter upside down or right side up? Is this a consonant or a vowel? It's like, so you can make discriminations and you see brain areas activating. And so they've been labeled, those are your free will areas, right? It's like, huh, are they really? Other areas, we've talked a little bit about default mode, right? And that's an area that's present at rest. So there's reasonably good evidence from imaging studies in people that are discovered to be misdiagnosed as vegetative or comatose, that seeing that the default mode network is functioning is a good index of that. And it's been associated strongly with sense of self-awareness and of social cognition, right? So I'd say if you had to pick an area that was most representative of self-awareness and situational awareness, like what is your surround, you should be thinking about the default mode network as being involved in that.
Default mode network doesn't sound like an area to me. It sounds like a collection of areas or something, a circuit.
It is a network. It's not an area. It's a group of areas. Posterior parietal, posterior cingulate are strongly linked. They're discrete areas, but they're strongly linked. They're also linked to medial temporal lobe, so episodic memory, and to subgenual cingulate and some dorsal cingulate.
And when you look for them in a patient, like if you're trying to figure out if they're really vegetative state or not, how would you see the linkage between these places?
They co-fire.
Okay, so you're looking at temporal correlation of activity.
Yeah.
And that's what basically defines the network under those circumstances.
In some of the networks that we looked at earlier, that was data that was acquired over, say, 15 or 20 minutes in each individual. And you're looking at every voxel, like 300,000 voxels, and you're looking to group them by their temporal coherence. So that algorithm doesn't care whether they're near each other or far away. It's not spatial local proximity, but you saw that they made blobs, right?
Yeah.
So regions that are talking to each other tend to be near each other, but then other regions, so you'll see that one area talks to another collectively, and so there are different groups. And so the ones that are most prominent when you're not doing another task, when you're just left to your own, are called default mode because they turn on when you're not doing something else. That's why they're default. Right?
So I was thinking, if I get back to my original question, I was thinking about some of these, like psychiatric disorder or a neurological disorder that doesn't have any obvious pathology. It's possible that the pathology could be connectional.
Yeah.
Like these brain regions are perfectly normal. It's just that they're not connected together with each other in the normal way. And is there any evidence that there's such a thing? And if so, can it be detected this way?
Not very
the
well.
class would be like autism or something like that, right?
Yes, autism would be a good candidate.
As a neurodevelopmental.
Yeah, for a neurodevelopmental disconnection syndrome.
You're asking probably about adult disorders that are not even obvious what change there is.
Yeah, that's what I was asking, but
Yeah,
I just was super happy
rerouting.
to think about developmental disorders too.
Yeah. So one technique that's been used a lot is voxel-based morphometry, right? So that's using structural imaging to look for abnormalities that are too subtle to see with your naked eye. If you show it to a radiologist, you'll say that's normal. But if you collect enough individuals with a disorder and without the disorder, and you contrast them, so you put them all in a common space, you pool all the individuals with schizophrenia and all the individuals without, and then you contrast them, do you see structural abnormalities? Yes, very clearly.
And they're located in particular places in the brain.
Yeah, they're
very
located in
consistent.
particular places in the brain.
And they weren't noted prior to these type of studies, though,
No.
right? Because it's always been very iffy if there were structural changes. And
so before this type of study, people had made reasonable guesses about where to look. Hippocampus is everybody's favorite, right? And so they would sample that and measure whether hippocampus shrinks if you have depression, right, or not,
Yeah.
right? And so there was evidence like that, which shows that yes.
But schizophrenia was less clear.
Yes. And so these studies are showing that there are abnormalities. The strongest are in schizophrenia and bipolar. And there's been nice recent genetic evidence that those are strongly linked genetically. And then the other disorders, substance use disorders, are showing abnormalities. The abnormalities in depression and in PTSD are less marked. But the strategy we're applying is to identify the areas where there are reliable structural abnormalities and look for connections between them and to see if the connections are disrupted. If you go with a premise that there's no other regional abnormality, it's just connectional, then you have to look everywhere and try and find where the connectivity abnormalities are. And that becomes a not very well posed problem.
But if you're looking for the structural abnormality and you see something in one part of the brain and you know that that's normally part of a network with another part of the brain,
yes,
you could look there.
Yes.
And if you do that, do you tend to get clusters of connected
Yes.
places that show the pathology? Or
yeah, that's pretty much the state of where people are now, is trying to figure out if we're looking in these canonical networks or if we're looking at regions of disease effect in psychiatric disorders, do we then find connectivity abnormalities? And the answer is yes, but the studies don't all agree with one another. And so we're kind of in the sorting it all out process. And I think that'll continue for a while. As people think of better techniques, then our results will gradually gel. But right now, I'd say, yeah, there's emerging evidence of this, and yeah, there's some very well cited, very well respected studies that do show that there's atrophy that emerges in psychiatric disorders, and that those are areas that are mutually connected. So it's network failure in these disorders.
when this is really refined and working well, do you expect it to be diagnostically beneficial? Because their diagnosis is often really difficult in some of these disorders, psychiatric disorders. There are arguments about diagnosis.
Yeah, there are arguments about diagnosis. I wouldn't count on the imaging to clarify the diagnoses, right? Because what's primarily observed is symptoms, right? And that's what if you have a disorder, you experience the symptoms, and the diagnosis is a clustering of those, which is somewhat artificial. And so people with one disorder may exhibit symptoms that are characteristic of another disorder. So people with substance use disorder may have depressive features. People with PTSD may have depressive features, but they may also treat their symptoms with alcohol or with other substances. And so is it likely that we're going to have pure psychiatric disorders, or we're going to have crossovers? And so thinking that the imaging will clarify how we categorize psychiatric disorders might be expecting too much.
Because it may not be a one-to-one mapping between the brain.
Might be a better mapping between symptoms and circuits that are associated with symptoms. The way that certain brain areas and certain networks produce speech, that's multiple regions working together. Certain brain areas and working together may produce depression, they may produce mania, they may produce different, sleeplessness, they may produce different types of syndrome or symptom. And then the different symptoms we collectively group into different disorders.
Uh-huh.
And so the imaging, I would expect, might relate better to symptoms.
So if you learned, forgive me for where I'm going, but
Yeah.
if you learned the brain areas that are altered that go with each symptom,
Yeah.
and then you discovered, oh, this patient has this cluster of symptoms and it has this cluster of brain areas. And now you decide, I'm going to try to design treatments. Then basically you'd be designing treatments for symptoms, because you'd go after that brain area that's responsible for that symptom. And then, oh, you've got this other symptom and we've got this other treatment for that one. Is that sort of the way you envision it working?
Yes. However, you should be aware that there have been published studies of psychiatric practice and how psychiatrists actually treat people. They treat them by symptoms.
Uh-huh.
And as they prescribe drugs, they pick a drug that's known to relate to, like if you have sleeplessness, if you have sadness, like whatever, you have overactivity, you have underactivity, they will choose drugs that they believe will remediate the symptom. And they're not driven primarily by diagnosis. So that is not what we're describing, is how psychiatrists already...
That's how it already works.
That's how it already works. Yeah, that's how it works.
But your information, if it applied clinically, could refine things, or won't have much of a dent?
As we move toward treatments that are neuromodulatory, then knowing the circuit, I think, will be extremely important. If we're working strictly with behavioral treatments or pharmacologic treatments, I think knowing the circuitry would be less important.
How will you use a circuit? How would you be able to treat a circuit?
So
is that like stimulation or?
Yeah, stimulation techniques like deep brain stimulation is a stimulation technique. Transcranial magnetic stimulation is a stimulation technique. There's direct current stimulation. So those different ways of stimulating will repetitively fire a circuit. Famous Canadian psychologist Donald Hebb, who emphasized the importance of circuitry and of retraining, saying that neurons that fire together will wire together, right? And that is how they strengthen circuitry over time. That's why motor practice, as you acquire motor skill, as you learn a sport, what are you doing? It's assumed to be synaptic plasticity and creating increased connections between brain areas. And so trying to modulate circuits is generally referred to as neuroplasticity, right? So you're trying to modify the circuits. And so understanding circuit structure at that level, I think, will become progressively more important.
Okay, well, thank you, Peter. That's been great. Very illuminating, stimulating.
Yeah, it's very stimulating.
And George?
Yes, thank you. Very
This has been Neuroscientists Talk Shop.